All right. All right, so I'd like to welcome everybody today to the December version of Micro Seminar. This is the last one for the decade. I think we can semantically argue that, but I think it's the last one of the decade. We're hoping to have many more in the next decade going forward. And again, if you have any intention or if you have any desire or want to share your science with us, just please let me, Dave Walters know, Cam Thrash, or Lizzie Wilbanks, let any of us three moderators know. You can email us, tweet at us, whatever you need to do. We will try to get you on the schedule, and anybody at any, I don't want to say age groups, any position in research. So grad students, postdocs, faculty, undergrads, if you want to share your research, definitely feel free to get in touch with us. So it's my absolute pleasure today to introduce one of my good friends in science. We're kind of in the same scientific cohort. We came up through the system together a little bit. So it's awesome to have Heather on today. Heather Hendrickson was an undergrad at the University of Utah. I was one of the most famous, or one of the, I'm going to say one of the people in microbial genetics who's left the biggest legacy, I think, in certainly the last couple of decades. So she was an undergrad working in John Roth's laboratory. And then after that, she went to grad school at the University of Pittsburgh with Jeffrey Lawrence, where she actually met up with somebody who I shared a bay with as a postdoc. So Heather and Emily Fisher we went to Pittsburgh together, and I kind of knew them both independently in a weird way. So she got her PhD at the University of Pittsburgh with Jeffrey Lawrence. And then after that, started as a Human Frontiers in Science postdoc fellow at the University of Oxford, and then worked a little bit with Paul Rainey in his lab as well at the University of Oxford. After that, she was fortunate enough to be able to start her own group at the University of Massey in New Zealand. So she's coming to us from the other hemisphere, the other side of the world, another season. Yeah, this is the magic of internet seminars here. So completely other sides of the world, and we're bringing you guys some science today. So Heather is her research is pretty all encompassing in a variety of different ways. She's very active in terms of SciComm. She's very active in terms of undergraduates and trying to bring people into science with using things like phage hunters. And then she also has a lot of cool research on things like cell shape evolution and a variety of other things that she may touch on, may not, but her research is in a variety of different directions, and it's all incredible. So I'd like to thank you, first of all, Heather, for joining us today, and I'm going to pass it off to you. So it's my absolute pleasure to introduce Heather Hendrickson.
Thanks, Dave. Thanks for the invitation to give one of these seminars. This is really exciting. I'll make one correction, so I actually moved to New Zealand to work with Paul Rainey here at Massey University, and then managed to start a laboratory in the neighboring institute. So that was how I first got involved in the research that I'm going to be talking about today. So to kind of introduce, so here we are. So what I'm going to be talking about today is exploring the evolution of spherical cell shape using experimental evolution. And this, I will try to acknowledge the people that were involved in starting the project and also contributing to the project as we go along. Yes, so to start, I will say, I am in New Zealand. So Massey University is in Auckland, New Zealand. New Zealand is often left off of maps. So just in case you're not sure, we're out here in the Pacific Ocean. We are not part of Australia. We are a separate country. So this is just what New Zealand kind of looks like. There's the North Island and the South Island. My university is actually spread across the North Island. So I'm up here in Auckland. And Palmerston North is where the university started, and we also have a branch in Wellington. And Auckland is the largest city in New Zealand, and we have probably close to 2 million people in Auckland. So this is a little picture of what our kind of main campus looks like here in Auckland, just to give you an idea of what the scene is. This is a beautiful picture from New Zealand. So we have, this is in Fiordland and the South Island where we have glaciers. We also have volcanoes, which has been a thing. So, so this is one of the volcanoes in the North Island, maybe a few hours from here. We're also surrounded by a lot of really beautiful bush, including fern trees, which are amazing. We have a lot of beaches. This is a beach in the Coromandel, which is on the East Coast. And we also have a whole wine island, an island off the coast of Auckland. This is a picture of Auckland, and this is the wine island off the coast. And this is a picture of Auckland, the city where I live. We also have healthcare and a really awesome prime minister named Jacinda Ardern. So that's a little taste of what New Zealand is. I've loved living here, and I've loved also associating with our experimental evolution and microbiology faculty. We have Sebastian Schmeier, who does bioinformatics, and each one of many of these faculty are involved in one way or another in experimental evolution and also microbiology. And I'll make special notice of Elizabeth Ostrowski does social amoeba dictyostelium research, and she's looking for PhDs in technician. Honor McCann does incredible work with plants and plant pathogens, and she's about to move to Max Planck in one of the Max Planck Institutes in Germany, and she's looking for friends to join her there. And Zhuzhan Zhang here is also looking for PhD students. So if you're thinking about your next move, consider New Zealand. I have loved it here. I've been here for about 8 years. All right. Bacterial cell shape diversity. I like to start thinking about bacterial cell shape diversity by talking about this wonderful quote from Kevin Young in 2006, who said, "To be brutally honest, few people care that bacteria have different shapes, which is a shame because the bacteria seem to care very much." And the reason that he says this, and this is also from this wonderful review, is that bacteria have an amazing array of different shapes that they take in nature. And this helps them with all sorts of kinds of issues that bacteria have. Issues that we'll talk about a little bit more later, but just the sheer diversity of different shapes in the microbial world is kind of staggering when you think about it. And many of these shapes we rarely consider or think about, but these are all ways that bacteria have adapted to their various environments. And perhaps unfortunately, but necessarily, today I'm talking about a very small number of those amazing shapes. And that is, I'm going to be talking primarily about vaguely rod-shaped bacteria and spherical bacteria. And one of the things that really surprised me when I first got into this field was thinking about just how bacteria have become rod-like and how they've become spherical. This is a 16S ribosomal RNA showing a large number, over 250 different bacterial species based on their 16S ribosomal RNAs. And the thing that's really surprising, I think, is that we think that bacteria is some kind of pre-bacterial organisms, were possibly kind of amorphous in shape and maybe membrane bound, but at the point where they hit upon having peptidoglycan cell walls, possibly, they also ancestrally were rod-like. And this is based on this kind of argument that says that, if you look down at the deep branches of bacterial families, you'll find that it looks like they were all rod-like, and that shapes like being helical or shapes like being spherical are actually derived traits from this ancestral position of being a rod-like cell. So each one of these red branches represents a point where we can see that the extant organisms in this group are spherical, and we can see that they are nested within larger groups that were all rod-like. So each one of these big red dots represents a point where an independent evolution from rod-like to spherical shape has taken place. And there are over 25 of these. I think my most recent count is closer to 27. And you can find that in our biochemical transactions paper, more details about this tree and what the species are. But it's really amazing to think about how rods have become spherical over 25 times in the phylogenetic tree. So if we zoom in on just a subset of those, we can see that there is a single gene that for many years has been recognized as probably very important in this process of going from rod-like shape to spherical shape. So now in these green branches, we have the bacteria that have become spherical, and we can see that there's a strong correlation, at least in this group of organisms that we're looking at here, between being rod-like and having MreB and being spherical and not having that gene any longer. So what is MreB? MreB is actually the ancient actin homologue in bacteria. So there's a strong structural correlation between actin and MreB, but we can't actually do a very good job of overlaying these in terms of sequence identity. The sequence identity is low, but structurally, this appears to be the ancient actin homologue. And many, many years of work by many laboratories have suggested that what MreB does in bacterial cells is it forms a filament along the inner membrane. And that filament is associated with what's called the rod complex for kind of rod-like cells. And this is associated with proteins that are able to build the peptidoglycan cell wall. So that class of enzymes is called the penicillin binding proteins because they're also the target of penicillin. So this is a two-domain enzyme that's able both to do the transglycosylation and the transpeptidation that's required for making the bacterial cell wall. And MreB is called the foreman of lateral cell wall construction because it places these at the place where they need to be in order to do elongation cell wall synthesis. So each one of these represents where an MreB might be present. And it's also been found that MreB exerts a slight inward force on the cell wall as it does that. And MreB orients itself according to the curvature of the cell wall. So in experiments where we have taken kind of model organisms and deleted MreB or interfered with it using a drug called A22, we can see that rod-like cells, when they don't have MreB polymerizing and acting as this foreman, these cells become spherical. But unless we put them in media that basically is able to kind of contain basically the guts of the cell, we tend to see cell lysis in model organisms. So if MreB is lost during this process of spherical cell evolution, how does this take place if MreB can't be deleted? Paul Rainey's group many years ago recognized that in the organism that they generally use for their adaptation experiments, Pseudomonas fluorescens SBW25, this was an organism that appeared kind of uniquely tolerant of an MreB deletion, and that was either a transposition event or a complete deletion. And what they observed was that the MreB cells are spherical and they appear to be viable. But that viability can be measured in two different ways. If we do just a simple optical density over 48 hours, we can see that the wild type cells, the rod-like cells, grow very well, and that the MreB cells are kind of chucking along, not doing quite as well, not reaching the same kinds of optical densities, but these cells appear to be able to go through both exponential and stationary phase. And if we put the population of MreB deletion cells in a direct pairwise competition experiment in a single flask with the wild type cells, and we measure the frequency of those two cells 24 hours later, we can see that the relative fitness of the rod-like cells is set at 1. So that's the wild type cells, and that these MreB deletion cells have a lower relative fitness in comparison. So they have a relative fitness of about 0.78. So that means that we have a viable population of MreB deletion cells, and Paul Rainey and his postdoc Monica Girth decided to do an experimental evolution experiment where essentially this is a bit like the Lenski experiment or other types of experimental evolution you might have heard of. They moved a subset of the cells each day after 24 hours of growth. So they are able to grow and divide and compete only against other MreB deletion cells. And they did this for about 1,000 generations in 10 separate lineages. And so where we came into the project, and this is my PhD student Richard Yulo's work, if you then take those evolved MreB deletion cells and just do an optical density, you can see that they're growing really on par with the wild type cells. So these are, again, the MreB deletion cells. They've really massively recovered their ability to grow. All right, so now we have a single strain where we can see a wild type, we can see an MreB deletion, and we can see these evolved cells. And if we look at them by electron microscopy, we can see that these cells didn't recover rod-like shape in the course of their evolution. They've actually taken that spherical cell shape, figured out how to be small, and I'll show you that they're also pretty fit. So not only can they grow, but they're pretty fit compared to the wild type cells, and much more fit than the MreB deletion cells. All right, this is a compactness plot, and what this allows us to see is a compactness of 1 is an approximately spherical cell. So these are cells that have been taken out of a liquid culture and then put on underneath a microscope, and this is the kind of the shadow or the what they look like under phase contrast microscopy. And what we can do here is we can plot their compactness compared to their cell volume. And in the wild type cells, we have this nice, typical organized increase in volume that's associated with an increase in elongation. So these cells are getting less compact. And this is what we kind of expect in most cell types, to see this kind of orderly relationship. In the MreB deletion cells, we can see that that relationship has really been lost. And so here we have cells that are maybe trying to divide when they have a massively increased volume compared to the wild type cells, and their volume just increases and increases. And so sometimes they're trying to divide at this point, but they've lost that orderly relationship. And if we take a look at the evolved cells, and this is one example of an evolved cell population, we can see that we've seen a restoration of that relationship between roughly volume and the point at which they attempt to divide. So we're really seeing a new feature in this population, that they've restored this orderly relationship. All right, so we can see that the transition from being rod-like to being spherical, we can see that with just the loss of MreB, we get poor growth and large volume and low competitive fitness. And if we look at that next step, we want to know what has happened in these populations of cells in order to allow them to go from this unfit large cell state to being something that looks a bit more like spherical cells that have evolved, that we're familiar with. And we also want to know if this is a good model. And the third question that we'll get to is, why would this kind of spherical cell shape evolve? And much of this work that I'm about to tell you about is published in a bioarchive, and we're just finishing the last experiments to resubmit that. Okay, so if we go back to the freezer and pull out the steps that we saw in this evolution of these 10 different populations, we can take each of those populations separately and do the same experiment again, where we compete them against the wild type and look at how well they do. And we started off, of course, with the MreB deletion alone, and that had a relatively low fitness. But we can see even after 50 generations, and especially after 250 generations, we've seen a massive increase in the competitive fitness of these mixed populations of cells. Some of them are doing very well compared to the wild type ancestor, and some of them are doing less well, but they've generally gotten there by 250 generations, which is pretty amazing. If we sequence the populations at 1,000 generations and look for mutations that have risen to a high level of frequency in those populations, there are a few mutations that really stand out, and one of those is this Pbpla gene is mutated in different ways at different locations in multiple populations. And there was actually another population down here where we could see two different Pbpla mutations were clonally competing against one another. So neither one of them completely overwhelmed the population, but we're seeing in multiple instances mutations in this particular protein. And it's another penicillin binding protein called 1A. So this is a crystal structure of penicillin binding protein 1A, and this is a diagram of that same gene. And what we can see is that it has 3 different domains. This is a set of organisms where this OB domain exists and its function is not completely understood. but this is the transglycosylase domain that makes the long chains, the long chains in the cell wall, and then the transpeptidase domain is responsible for linking the chains together. And then there's this OB domain. So we decided, and I should point out, the active site residues for penicillin binding or for that transpeptidase are here, and we can see that this mutation number 1 from line 1 is very close to that site. And we also decided to focus on this OB domain mutation that we found in line 4. So I will refer to these either as the line 1 mutation or the line 4 mutation, but hopefully I'll remember to say the OB domain mutation and the transpeptidase domain mutation. So we wanted to understand a little bit more about why these particular mutations were coming up. And of course, part of that is understanding what is the relationship between the Pbps that are associated with MreB and this other Pbp. And some very beautiful work by the Ethan Garner's lab at Harvard has looked at how these different penicillin binding proteins move relative to one another. And what they have found is that the activity of this set of cell wall building enzymes is generally taking place in association with the direction that it's given by the MreB. So the MreB gives it this structure, and they are generally acting in this kind of orderly way across the cell wall. And those MreB filaments are kind of directing the activity of this enzyme. But if we look at fluorescently how these Pbp1As are moving, in that system, they appear to have kind of a more random movement. And it's been suggested that they might be making kind of more, random associations, but also doing the same sort of cell wall synthesis activity. And some people have suggested that these are affecting one another, and some people have suggested that they're acting completely independently. So that's some beautiful work from that laboratory. And of course, this is the Pbp1A that we're interested in, and its mutations. All right, so we want to investigate why these Pbp1A mutations are having this effect of being able to take these large spherical cells and then transitioning them to these small spherical cells in order to understand how spherical shape evolution might take place. So in order to investigate this, we want to reconstruct the Pbp1A mutations in both backgrounds, the MreB deletion background and the wild type background, and take a look at what this does to the cells. Similarly, we want to take a look at these Pbp1A mutations and their effect on cell wall construction by using fluorescent analogs of the cell wall materials. And last, we'll look at the Pbp1A mutations and how they actually affect activity, again, using a different type of fluorescent analog and a different type of experiment. So those are the 3 experiments I'm going to take you through. All right, so first, the reconstructions. So if we just take a look at the Pbp1A mutations, either that transpeptidase domain mutation or the OB domain mutation, and we put them into the wild type cells without the MreB deletion, we don't see a significant change in the overall shape of the cells. But if we take the measurements of these cells, we actually observe that there is, these cells are much thinner. So it's not completely obvious from this kind of image, but if you measure them and put them on this kind of compactness plot, we can see a significant difference in the thinness of these cells. So that suggests to us that the Pbp1A mutations are doing something that's relatively subtle in the wild type cells. But if we take just those 2 mutations and we construct them in the MreB deletion background, we see that this is sufficient, the single mutation is sufficient to remake these small spherical cells. And we've also done fitness assays and seen that these are enough to increase the fitness of these cells. So that's our first hints about what these mutations might be doing. But in order to see how they're affecting the cell wall construction, we wanted a way of visualizing what's actually happening at the cell wall. And this is enabled by these beautiful fluorescent D amino acids that are being produced. And we were able to obtain some of these. So what we do is we flush the bacterial cells with these fluorescent D-amino acids and then wash the D-amino acids away and look at, in the time that we exposed them, and I think it was like 20 minutes, where did these fluorescent D-amino acids get incorporated into the cell wall? And generally, septation is one of the places where a completely separate set of guided PBP activity is taking place. This is guided by the FTSZ or FTSZ. And then the elongation synthesis in a wild type cell is this kind of distributed and smooth activity that's the product of both the MreB associated PBP1As and the non-MreB associated the MreB associated PBPs and the non-associated with MreB PBP1A. So this smooth signal of where the amino acids are being incorporated into the cell wall is what we're interested in looking at first in the MreB deletion. So what takes place in the cells in terms of cell wall synthesis when we delete MreB? And we were somewhat surprised by this because what we see is that when FTSZ is associated, we still see some of this managing to make a septum. So we can see some of that FTSZ associated cell wall synthesis, but the elongation synthesis is really very disrupted. So what we're seeing in the absence of MreB is these patches and clumps of cell wall synthesis that are just taking place all over these cells. So the loss of MreB is really probably, we would guess that this is primarily the product of these PBP1As, and that the loss of MreB has led to the disorganization and the loss of that beautiful, smooth signal of cell wall synthesis. So we'd like to take a look at what happens in these cells if we then put in either the transpeptidase mutation in the Pbp1A or the OB domain mutation in the Pbp1A. And what we see in both of those cases is, again, these smaller cells, but these smaller cells have now apparently regained what looks like a smooth cell wall synthesis. So that disorganization is really lost when we lose, when we have mutations in the Pbp1A. And this suggests to us that now we're seeing mostly FTSZ mediated septum based cell wall synthesis. And this makes us suspect that these mutations are loss of function mutations in these Pbp1As that are more distributed in the cell. So what we did then in order to get a more precise and a more specific look at what the activity is of the Pbp1A specifically is we engaged with Felipe Cava in Sweden. And these are preliminary results where we look at Bocillin, which is a fluorescent penicillin analog. So Bocillin should be binding at this transpeptidase site. And if you take the wild type cells, you see that there are lots of different enzymes that have a little bit of binding to the Bocillin. So we know that there are lots of different flavors of PBPs in these cells. So there are some that only do transpeptidase, there are some that only do transglycosylase. So in the wild type, we see all of these different bands, but in the Pbp1A deletion, we see the loss of this very specific band. And in the wild type with the transpeptidase domain or either the OB domain mutations, we also fail to see this band. So we're in the process of constructing the Pbp1A MreB deletion, Pbp1A deletion, MreB deletion double mutants so that we can test a complete set of these. So this is ongoing work, but this really gives us a picture that suggests that these mutations are actually eliminating this transpeptidase activity, and that even maybe the OB domain is eliminating this transpeptidase activity. All right, so that leads us to a summary of those results. So when we reconstruct the mutations in either of these backgrounds, we see that we get a reduction in cell size in the MreB deletion and a reduction in cell width in the wild type. The Pbp1a mutations have an effect on the cell wall construction as well. So in the presence of the Pbp1a mutations, we see a decrease in that disorganized construction of the cell wall. And last but not least, it looks like the Pbp1a mutations inactivate the cell wall transpeptidation in either one of those mutations. This leads us to something of a model. So in a normal wild type rod-like cell, we have cell wall activity that's being negotiated by the FTSZ associated PBPs, and the combination of MreB, which has this tendency to kind of constrict the cell, and the Pbp1as, when they're mutated, appear to allow the cell to get much larger, and they engage in the absence of the MreB associated PBPs with just this very disorganized kind of almost frenetic sort of cell wall construction. But the FTSZ associated activity is still present. And then when we get mutations in the Pbp1as, we get a restoration of this kind of orderly cell wall synthesis that's primarily taking place at the FTSZ, but there are other unknown PBPs that are still contributing to cell wall synthesis once we have mutations in the Pbp1a. All right, and this is in terms of thinking about how spherical cells have evolved, this is kind of satisfying in a way that we might not have anticipated, but maybe we should have. And that is that in rod-like cells, we see elongation synthesis, and we also see FTSZ-associated synthesis. But in organisms like Streptococcus or Staph aureus, that are kind of naturally evolved spherical cells, most of the cell wall synthesis is believed to be really taking place at the FTSZ. So we're seeing a recapitulation of this type of cell wall synthesis in extant spherical lineages. We're seeing a recapitulation of that in our spherical evolution cells. And this is also kind of substantiated to a degree if we look back at that tree that I showed you earlier and the presence and absence of MreB in these rod-like and spherical cells. We also see a tendency towards loss of PBPs in these organisms. So rod-like cells on average have about 8 PBPs, and spherical cells or round cells on average have around 3.6 PBPs. So this suggests to us that we're seeing something in our experimental evolution that's relevant to the evolution of cell shape in naturally evolved cells. All right, so far what I've talked about is the loss of MreB in rod-like cells and how this leads to these large spherical cells, and that if we then evolve those cells, we observe Pbp1a mutations as one of the primary mutations that come up, and this appears to balance the cell wall synthesis. But the last thing that I'd like to talk about is a little bit about why this transition would ever happen at all. So rod-like cells appear to have become spherical cells many, many times in the phylogenetic tree that I showed you in the beginning of the seminar. But the story that I've told you should seem a little bit strange, and that is that if we look at the competitive fitness of each of these different states, if MreB is a transition towards becoming, if the MreB deletion is a transition towards becoming a small spherical cell, what I've suggested is that there's a decrease in fitness in order to get over to that state. And of course, this is a decrease in the competitive fitness against the wild type cells that we observe in liquid culture. But it's sort of a strange story. So why would this have happened so many times in nature if there's this state that is kind of not very fit on the way to becoming a fit spherical cell? And this kind of brings us back to why cell shape is important to bacteria in the first place. So we can see that the shape of a cell is going to be one of the primary traits that mediates a lot of its important interactions in nature. So if bacteria rely on their cell surface in order to acquire nutrients and to get rid of waste, having a spherical cell shape has actually decreased that surface area compared to your volume. So that seems a little bit strange. Obviously, traits like predation are going to affect cell shape. But if you think about motility or surface attachment, or even the symmetry that bacteria use in order to segregate their DNA, in many of these cases, going from rod-like cell shape to spherical cell shape does not necessarily seem like an advantage. In addition, we know that bacteria actively regulate their cell shapes. So here's an example of a bacterium that's becoming spherical, and then it's getting a bit larger and more elongated. And we know that they do this in response to the presence of nutrients in the media. Higher nutrients generally leads to larger cells. We know that bacteria do this when they're faced with predation or immune system challenges. And we also know that things like stress or DNA damage leads to changes in the shapes of the cells. For example, they might start elongating. So if we look at what we expect in terms of the transition from rod-like to spherical cell shape, where would this be an advantage? These are cases where it doesn't look like this is advantageous. There's been some beautiful work by Sam Smith and Kevin Foster at Oxford recently in the last couple of years, where they suggested that spherical cells position themselves differently just by being the shape that they are, that they tend to go to the tops of biofilms. So maybe biofilm location is a potential advantage for spherical cell shape. But what really drew our eye was this predation that shows up, and to a lesser degree, maybe immune system challenge. So we wanted to investigate the effect that predation might have on this evolutionary transition, and specifically these MreB deletion cells. So we decided to pursue a project where we wanted to look at what was the significance of amoeboid predation on spherical cell shape. So amoeba, of course, are single-celled eukaryotes. They are, most of the time, they are shapeless and crawl around on surfaces. They eat bacteria by phagocytosing them or engulfing them. And they often have multiple forms. If they run out of food, for example, they have cyst-like forms or spores. And according to some of the older literature, 60% of bacteria are killed each day in the soil by amoeba. So this suggests that an amoeboid predation might be a major effect on the ecology of bacteria and in natural spaces. So this is a view of some of the amoeboid predators that we have in our laboratory. This has been sped up a little bit, as you can see, but these are really the wolves of the microbial world. They just kind of rip through bacterial cells and engulf large numbers of them and just keep munching on them. And they're really a pretty impressive organism to have in the laboratory. So this is Naegleria gruberi, just munching away on some rod-like cells. So we considered the idea that in our wild-type cells, they have about a 1x volume, and that if you actually measure the volumes of these MreB deletion cells, on average, they have about 6 times the volume of a wild-type cell. And the volume tracks with the DNA concentration. So these guys have lots of copies of their chromosomes. They are not very good at handling that, so that's probably part of the division issue that they are having. But when we see these spherical cells, they have basically restored that 1x volume. So they go from 1x to 2x and divide. And this put us in mind of a lot of the ecology literature. It turns out that amoeboid predators prefer medium-sized cells. So they are, this is called Goldilocks predation. So if you have a large population of cells with different sizes, they tend, after grazing, to go after whatever the medium-sized cells are, and small cells or large cells are generally going to be ignored. And so we thought maybe if we looked at the preference of protozoan predators, we would see that the MreB deletion cells might have an advantage in the presence of protozoan predators. So this is work that was done by my master's student, Danielle, who's now a PhD student with me working on bacteriophages. And the suggestion for her master's project was that first she would find some protozoan predators and then test their preference for these cells that differ in their size or their volume. And the hypothesis was that the MreB deletion cells might actually do better than rod-like cells under protozoan predation. So Danielle's first task was to get a hold of some amoeboid predators. She managed to find in our own backyard here in New Zealand some dictosteliids. These are probably Dictostelium giganteum, but you can see they have this fascinating life cycle where they're basically running around as little amoeba, and then at some point they send out signals to one another that they've run out of food, and they form what's basically a cooperative multicellular slug. This slug is then able to crawl up to a surface, and they make these beautiful structures called sori. And in the top of the sori are spores that are going to enable that population of amoeba, the ones that made it into the spores, to then spread to a new location and start propagating as amoeba again. So these guys are really cool, and we found some here in New Zealand. We also obtained some Naegleria gruberi from some colleagues in Pittsburgh, and these are amoeba that have a kind of amoeboid predation form like I showed you before. They also have a form where they swim around, and they have cyst-like forms as well. All right, so the experiments for testing the preferences of these different predators is pretty simple. We do pairwise line tests. So these are lines of bacteria, and we put maybe 20,000 amoeba on a paper disc in the center of the cell, and then we measure over time to see what is the frequency, or what's the rate of predation down this line. And then we adjust for the cell density of the different cells in order to come up with a feeding preference or a predation preference. So what we see is that there are massive differences in the preference of these predators for these different types of cells. This is our evolved line 1, I think. So they do not like to eat the MreB deletion cells. They have an 8-fold preference for the wild type cells in the case of Naegleria gruberi, and about a 4-fold preference for the evolved cells over the MreB deletion cells. And we see the same kind of pattern with the dictosteliid cells, which are slightly smaller. Is that right? Yeah. So if we put this into relative preference, we can see that they have a low preference for the MreB cells. The evolved cells are doing better than the wild type cells, but the wild type cells are really munched. And we see the same thing with the dictosteliids. So there's a real preference for eating these wild type cells, and so they have a low fitness in the presence of the predators. So that's great, but what we want to understand is it possible? So if we have a population of wild type cells and we see an MreB deletion mutant evolve in the presence of predators, is this difference in preference sufficient so that the MreB deletion cells could actually grow into a sizable population where that secondary mutation that makes them both fit and not very preferred by the predators, is there really time for that kind of population to develop? And so in order to approach this, we decided to take an agent-based modeling approach. So Danielle was a double major in computer science and genetics. So she could make an agent-based model with the cooperation of one of our colleagues here, Daniel Plain. And so what they decided to do was to make an agent-based model where they would have wild type cells and predators. And if the predators don't find any food in a sufficient amount of time, they can become cysts, which are these dormant states. And then we had MreB deletion cells. And we wanted to start in kind of a reasonable evolutionary scenario with a single MreB deletion cells and maybe 100 wild type cells in this population. So we held MreB division to be a constant, so they're slow at dividing. And we could run different versions of this model where we could change the preference of the predators for either the wild type or the MreB. We could vary the concentration of the predators. And what we wanted to know was to what frequencies can we see the MreB deletion cells in these populations. This is what like a final model might look at like. You can see the cysts and you can see the little amoeba, and these are the MreB deletion cells. And the green is the rod-like cells. So when you run one of these models, this is what it might look like. So we can see, the MreB deletion cells initially are not doing very well. Some of the predators are managing to eat them, but as the predators start clearing out the rod-like cells, this actually gives the opportunity for these MreB deletion cells, even though they're slowly dividing, to start taking over a significant size of this surface. What was kind of cool about this is that we could also see that as cysts form, those end up being little hiding spots for the bacterial cells. So bacterial cells can survive if they're right next to a cyst. And that was actually something we observed in our plates as well, which was kind of cool. So the agent-based model really kind of directed us towards an observation that we hadn't necessarily anticipated. So we ran many of these agent-based models and looked at if we change the preference for wild type versus MreB. So this is, they have a 10-fold preference for the wild type cells over the MreB deletion cells. This is an even preference, so a 1-to-1 preference. They'll happily eat wild type or MreB. And this is the number of amoeba that we had in the initial run of the population. What we can see here is that we have a large space that we can observe in our agent-based model, where the MreB deletion cells are able to get from populations of 16% of the cells after 1,000 steps to up to 50% of the cells, depending on the fold preference and depending on the number of amoeba that are present in the agent-based model in the first instance. So this is kind of cool, and I'll just point out that this is where the dictosteliid kind of preference was kind of floating around 4, and the 8-fold preference is around here. So we can see that in these sort of reasonable models, we do see that there's an opportunity for as many as 30 or 32% of the cells or 40% of the cells to be MreB deletion cells after 1,000 steps. All right, so this means that instead of this, very simple idea about what the fitness of these cells would be based on only their pairwise competition and their ability to outcompete one another in liquid media, if we instead of this kind of assumed and kind of typical, relative fitness landscape, look at an ecological landscape in which protozoan predators might be driving the frequencies and the fitness of some of these organisms, we can see that the MreB deletion cells have a higher fitness than the wild type cells in the presence of protozoan predators, and that by the time they get these secondary mutations and become small spherical cells, not only can they now grow as well as the wild type cells, but they're less preferred by predators than the wild type cells. So we think that what we're seeing here is really that protozoan predation and the MreB deletion can be a ratchet that kind of sends a cell lineage towards having a spherical cell shape. And so we don't think that this is the only explanation for why spherical cells have become spherical after having had rod-like ancestors, but we think that this is an interesting potential hypothesis for how this kind of transition might happen in nature. All right, so what I've talked about is this idea that bacteria were ancestrally rod-like and that they've become spherical independently in many separate instances. If you lose MreB, which is what we think is part of this transition, cells have poor growth and large volume and low competitive fitness, but they can rapidly acquire mutations that restore balanced cell wall synthesis and make them into these small spherical cell shapes. Again, I don't know why spherical cell shapes has evolved, but I think that predation is an interesting and powerful hypothesis that has not been discussed in the literature in the past. And what has been discussed is biofilm placement or packing. And there are certainly stories that you can tell here also about antibiotic resistance and how that might have contributed to spherical cell shape as well. But I'm not going to get into that today. All right. So what I've told you about is the impact possibly that protozoan predators might have on this transition from rod-like to spherical cell shape. My laboratory is interested also in the relationship between bacteria that are completely harmless and how protozoan predators might be driving the evolution of virulence traits. And we've started recently a new project where we're interested in taking protozoan predators and bacterial prey and looking at whether or not we can involve new endosymbionts starting from this prey and predation state. So those are the kinds of things that we're doing in my laboratory. There are a ton of people that have been involved in this research. So this was, of course, started by Paul Rainey and Monica Girth. Felipe Cava is a very important collaborator. Zhizhan Zhang and Daniel Plain have been involved here at Massey. I have a great group of PhD students, and these are my new PhD students, and I'm collaborating with Elizabeth Ostrowski and Ant Poole. And our new lab member, Mike Miller, who's a research officer, are involved in the endosymbiosis project. So I have lots of people to acknowledge. I should also acknowledge that I'm funded by multiple different groups here in New Zealand. I'm involved in the Howard Hughes Medical Institute's phage hunt, phage hunt for undergraduate students, which is an amazing program. And we also have some phage work that's funded by the Sustainable Farming Fund here in New Zealand. And with that, I can take questions.
Awesome. Thank you much. I love that story. So just to remind everybody on the stream, so there's a few people watching actually, which is nice. If you have any questions, just tweet at MicroSeminar. You can even ask them in the chat of the YouTube. I'll kind of monitor there. I have a question though. So yes. I'm kind of biased and always thinking about trade-offs. So it strikes me that if you have Pbp1a and Pbp2a, like organized and not organized combination of building the cell wall, what's the point of having the disorganized way to do it? And then if you have the disorganized way, like what's the trade-off to, I guess, cell wall being able to make that when you do transition?
So I don't know, but the work that Ethan Gardner has been doing, he's done these interesting visualization techniques to see if you increase the amount of the MreB associated cell wall synthesis, that's kind of that orderly synthesis that's taking place in kind of that direction. You end up with thinner cells, which is consistent with the kinds of phenotypes that we're seeing. If you instead increase the amount of the Pbp1a associated synthesis, you end up with slightly larger cells. So there's this balancing act that's taking place between those two. But the other thing that he seemed to suggest from those kinds of visualization techniques when he increased the one or the other was that you're getting these, it looks like less, the Pbp1a mediated cell wall construction is making more curves, like there's more curves kind of interwoven in the cell wall. So instead of just having these straight lines, you have, I guess when I've been thinking about it, and I've been thinking about it in terms of like, if you are making a basket and you only make, one direction and you don't have anything kind of weaving those together, then you're not going to have as strong a basket as you would if you had these two different types of activities. So I haven't seen him, suggest that, but when I'm reading these papers about these two different types of kind of, squirrely synthesis versus this orderly synthesis, that's my kind of takeaway. So I think that it probably has to do with the strength of the cell walls, and having both of these activities is probably better. The other PBPs that I don't really talk about are also sort of doing, we're not really sure like where they're acting, and they may just be distributed all over the place. But then of course it's obvious that having that FTSZ or FTSZ oriented activity where that really brings down the septum is another important function that's completely separate.
Cool. And then do you see, so since peptidoglycan plays such a role in cell shape in certain strains or certain species and not others, like gram-positives, it's super important. Gram-negatives, it's important. Maybe not as important as in gram-positives, given kind of the way things are structured. Do you see any prevalence for biases in terms of who becomes spherical or who becomes, who stays rod-like?
I haven't. And there was a really beautiful paper a couple of years ago, and I'm not going to remember the authors right now, but it focused solely on that same kind of tree, but only for the firmicutes. So if you look at the firmicutes, what they were hoping was that they could see a relationship between traits like motility or traits like virulence or host association. And they tried to associate, the transition from rod-like to spherical cell shape with one of those different types of traits. And they ended up kind of coming up with like, we can't see a relationship between, the presence of, motility driving things like flagella. We don't see a relationship between host association. They kind of like went, I don't know. And they came up with something like, it's really good to be able to adapt or something like that. I mean, the conclusion of the paper was basically like, we thought we would see a strong relationship between one of these things and we really didn't. And again, for me, that looks like there's an ecological parameter that we are forgetting here. And maybe it's something like predation, maybe it's something like biofilm formation, or maybe it's a mix.
Cool. All right, so I'm going to take a look at the different streams and just see if anything pops up in terms of questions. YouTube, nothing. Not seeing anything on Twitter. So with that, I will just say thank you very much again. I'm going to end this now, but we'll chat for a couple minutes afterwards too. But thanks again for this. And again, I'll archive this on YouTube, so this will be up for whoever wants to watch it in the future. So thank you much. That was awesome.